So good morning, everyone. So I'm going to talk about stochastic simulation, and I have a few slides, but I'm actually going to, the slides are already on the drive. So you're welcome to download them and look at them. I am going to pass a few of them quicker than I wanted. In fact, I might skip a few because we are actually two people talking, and there's a lot of material to go through. So I'll try to do my best to give a very, very quick presentation. So yesterday, Laszlo already mentioned that there are issues with chemical kinetics when you get down to looking at specific molecules. And when you have very large numbers of molecules, we normally use continuous numbers to represent them, and we have concentrations and things like that. But if you get down to small numbers of molecules, you have to realize that you cannot have fractional numbers of molecules. So there's no such thing as 2.3 molecules. It's either 2 or it's 3. Or you cannot have 15.3. It's either 15 or 16. Fractions of a molecule are not molecules themselves, or if they are different molecules. So this has to be integers. And the problem is that differential equations are continuous, and they would definitely generate such numbers as 2.3, etc. So in reality, if we want to simulate these systems at a very microscopic level, we really have to use integers. The second thing is that the reaction events are also not continuous. So a reaction happens at a certain discrete time, and it basically at any specific time, the reaction either happens or it doesn't happen. And there's another thing which is we also saw Brownian motion, and the noise is intrinsic in these chemical systems, and so the noise effect it's also important to look at. And so these are all motivations to use stochastic kinetics. The final thing I would point out, and this is an interesting calculation that some of you might have done or not, but if you think about the volume of a cell, for example, an E. coli cell, which is, albeit a small cell, some eukaryotic cells are larger than that, but still, that cell has about a volume of a few femtoliters, like say 1.5 femtoliters. That's 10 to the minus 15 of a liter. And if you think about how many molecules is in a mole, and you can do the calculation that for something that is exists in the cell at the concentration of micromolar, 10 to the minus 6 of a molar, you basically can calculate how many molecules there are, and that number is 945. So that's a micromolar. So if you have something that is a nanomolar, then it's going to be 9 or 10 molecules. And that's of the entire E. coli cell. So this is just so to you to figure out that actually at the space of a cell, or say an organelle, say mitochondrion, which is probably about the similar size of bacterium, you have you can easily have very small numbers of molecules. So we move to stochastic dynamics, and so I'm just going to compare very quickly how we do rates of reactions in the normal chemical kinetics on this side, which is the differential equation approach, and on this side, the stochastic approach. So if you have a constant reaction, which is, you specify the rate of reaction is equal to a constant, in the stochastic approach, you have a propensity for the reaction to occur is also a constant. The propensity is an important concept. It's the probability that a reaction happens at a certain time. So it's a probability, but it's related with the concept of important to think about. It's not just a probability. It's a probability that something happens at that time. And that probability is also constant for all time if it's a constant rate of reaction. You can see the similarity already of the two rates. Now, if you have a first order mass action reaction, A is transformed into P in this case. The traditional mass action kinetics has the rating equal to A, the constant, times the concentration of A. And the propensity is proportional to the number of molecules of A. So the propensity here is a constant times the number of molecules. Again, see the similarity of the expression. Now to the second order reaction, you get that expression K times A times B, and the propensity is now proportional to the number of all AB pairs, and the number of AB pairs is just a product of A times B. Finally, there is a special case. This is also a second order reaction, now it's A plus A. So it's 2 molecules of A. And in normal chemical kinetics, you have K times A squared. In stochastic kinetics, you realize that it's proportional to the number of A pairs. So if you take one of the A molecules first, this one, then the second one that one is no longer available to be pairing with itself. It has to be a different one. So it's A minus 1. It's the total number minus 1. So it's slightly different than what we would have for the case of just second-order reactions. Now, you can calculate that there is even a correspondence between this C constant that I've been using and the K constants. So you can calculate C's from K's. The expressions include the Avogadro's number, of course, and the volume as well, the volume of reaction, to have the right unit. So this is very quickly to explain how we can convert automatically, which we can do in COPASI, a model from a deterministic ODE type approach to a stochastic Gillespie type model. So this is nothing, I mean, I'm just showing you this to show the actual probability density function. This is called the next reaction density function. This is the probability of a reaction happening at time tau and taking into account the molecule X. And this Gillespie derived an algorithm which is now widely used. It's a Monte Carlo technique that basically just draws from this distribution. It calculates what is the tau, that is the time that the next reaction happens, and which molecule, or sorry, which reaction J happens at that time. So essentially what that algorithm does is that it samples the times of reaction, and then it samples which of the reactions happens. And that's all based on the propensities that I mentioned before, which, if you remember, they are very well related with the rates of rate constants. So I'm going to skip this and just basically say that Gillespie derived this algorithm that basically calculates all the reaction propensities. It has to calculate the sum of their propensities as well. And then it generates 2 random numbers to calculate what is the time for the next reaction and what reaction index is the reaction that happens. And then it just increases the time by that value tau, and it makes the reaction happen. That is, if it was A transformed into B, it removes one number from A and it increases a number by 1 for B. And then it continues going on like this. Now, this tau value can be a very, very small number depending on the number of molecules. In the past, there are some papers to motivate you why we might want to use stochastic kinetics. The effect of stochastic kinetics sometimes is the same as not as in the deterministic cases Laszlo showed yesterday, but there are some cases where it isn't like that. If you go down to stochastic simulations, you see things that you would not see in the deterministic case. And this example of the lambda phage, the switch between lytic and lysogenic behaviors of the lambda phage, the famous paper from Adamarkin back from 1999. This is when there was put a big emphasis on the fact that sometimes you really need to do the stochastic simulations. And I leave this just as a motivation. I'm going to stop the presentation here, and I'm going to move to showing you how to using COPASI for this purpose. So I'm going to remove my simulation. Hopefully you're all seeing my screen. And this is also a good time to ask if anyone has questions. Looking at see if I see any hands raised. Okay, so looks like there's no questions. So I'm going to now run a model which I also have on the drive. So if you are using COPASI and you want to run it, it's called the Schlogel model. This is a model that is famous because it comes from the 1970s, and it is a bistable model. It has 2 possible steady states, and I'm going to show you a couple of things. First of all, I'm going to run the first simulation in the deterministic mode. So I'm going to use an ODE and just run the simulation. And you see here in my plot, let me rearrange my window a little bit to show the plot a bit bigger. This window can be smaller. so that I can make this plot larger. What I'm showing you here is the time course of this variable x, which is the model actually only has one variable. It has 3 species, A, B, and X, but X is the only variable. And it's basically a very simple reaction network. It has some autocatalytic reactions here. There's one autocatalytic reaction. So X depends on the initial concentration of X as well. So what I'm showing is that there are 2 possible steady states represented by the blue and the green line. This is the lower steady state, that is the upper steady state, this one here. And if I have a certain initial condition, the model converged now to the lower steady state. Now I'm going to show you a slider, which I'm going to get by pressing this button here. And the slider is something that allows me to change a parameter of the model and get automatically calculated the model right away. So I'm going to add a new slider for the time course. And I'm going to have the initial concentration. I'm going to add it just so you see. I'm going to add the initial concentration of that species X. Remember, the X is not a parameter of the model. It's a variable. It's an actual variable. So what I'm going to show to you is that depending on the initial value of X, we can go to the lower steady state or to the upper steady state. So I changed it to be 318, and now the model converges to the upper steady state. If I move it a little bit further down, it still see the initial condition here. It's now slightly lower. It still goes to the upper level. If I decrease it a little bit further, now it goes to the lower level. And I can continue doing this, and it's very deterministic. I know where there is the change between values, where it goes to one or where it goes to the other. This is the deterministic condition. So now I'm going to switch to a stochastic model. And in COPASI, to switch to stochastic, it's very simple. All you need to do is you need to switch the algorithm that you're going to use, the method for the integration. So I'm in the time course window, and here I'm going to change from deterministic to stochastic. And you can see there's a number of stochastic options. I'm going to choose the direct method. This is the method that is described by Gillespie. The Gibson-Brook is an alternative way of specifying that method, but it's only good for models that have many variables. Otherwise, the direct method is actually faster. So I'm going to choose the direct method, and I'm just going to run exactly the same thing. And what you saw now is that the plot is stochastic, right? So you see the number of molecules here, and it's now, it starts at 200 and something, and it's oscillating between a certain value. And in fact, the steady state here is really an oscillation around it. Now, we told you before that one stochastic simulation is not enough. You have to run several of them. So I'm just going to run another one again, and another one, so you can see. And there's actually a way of running many of them together in COPASI. We can repeat this simulation automatically, and that is done in the parameter scan window. So now I move to the parameter scan window, and in parameter scan, I'm going to show you how that works. Normally, you have to choose time course here, and I'm going to create a repeat. So I'm actually not going to scan any parameter. I'm just going to create a repeat of the same thing and a certain number of times. So I'm just going to click repeat and press the create button. I add here a repeat. The default is 10 values. I'm going to run 20. So I'm going to run 20 stochastic simulations, one after the other, and the plot will accumulate all of them. And that's the result. And you now might have seen a surprise here, because not only did you get several oscillations around the steady state, so you can see a steady state is really oscillations around it, but now you saw that some values converge to the upper level. And this is because of the stochasticity. So in the fluctuations, some numbers here made you switch to the upper steady state. And if I can run more than 20, I can run 100. And you will see the same thing. You'll see a few more. In this case, only one so far, 2, 3. So it's a rare event. It happens a rare number of times that it goes from the bottom one to the higher one. But maybe it's because of the value of my constant. So if I switch my constant a little bit higher, it was 170, now it's 180. It's still rare. But if I increase it a little bit higher, the initial condition, it starts to become more frequent to be on the upper side. And if I increase it even further, it becomes now more common to be on the upper level than on the lower level. And now if I move it quite a bit further up, the initial condition, now it's mostly on the upper level, but sometimes it might jump to the bottom. In fact, I might have switched it too high. And now the probability is very, very low. So it maybe out of 100, none of the times came down. So I'm going to just move it a little bit lower. And yes, so you see now the probability is small, but there is a probability that it will find the other steady state. It will jump to the other side. Also, if I have run a longer time course, which we can do, there will be a probability. So rather than doing 20 seconds, I'm going to run for maybe 200, or maybe let's say even 2000. Now in one single simulation, it's on the upper level, but it could be that at some time it will jump to the bottom one. Now this, I may have to make the constant a little bit lower, but or the time longer, because the probabilities of course depend with time. So there you saw it. So it was on the upper level and it switched to the lower level. And if I run the next time, it maybe doesn't switch, or it's always on the lower level, or now it's always on the upper level, but sometimes it can switch from one to the other. So I think I have maybe some hands raised perhaps. I'm going to give you time to ask questions. Please go ahead. Gamal, I think.
Good morning, Pedro.
Good morning.
In what specific instances do you see the utility of using stochastic versus deterministic methods? Like in this example, I'm looking at solid state physics for myself is extremely valuable, because molecules are, in my opinion, this is extremely powerful. But when do you see this in biological systems?
Well, I mean, one of the things I mentioned is when you see this, when you have concentrations that basically mean you have a number of molecules that is fairly small. Okay, so I can increase the size of this system, and if we increase the size of the system, you will see that this behavior becomes less probable. Right, so if I have more molecules, so I can do that by changing the initial concentration, the volume of the system. So I have.
But my question is, in what specific examples, in biological examples, will you see this?
Well, I mean, I showed you one motivation example. That is mostly things related with gene expression. In gene expression, you normally have some noise, and you have a low number of molecules. For once the gene number, usually it's only 2 copies. You may have some repeated, but it'll be a small number of copies. So that's already a small number. And the number of mRNA molecules produced is also normally small. So that's a situation that is commonly a case for using stochastic kinetics, yes.
Perfect, perfect. That's a very good example, yes.
Any other questions?
Hello.
Hello, yes.
Hi, good morning. I was wondering whether there is any value of X for which one of the 2 steady states cannot be reached, is excluded from the stochastic, like any value other than 0?
I'm not sure of that, but that is something that I'm not sure. I mean, this system, this particular model is very well studied. So I think there might even be an analytical solution for that people might have studied the probability density function. I believe that there is a threshold at some point where you might show the probability is going to be smaller than feasible value, but that is something you can study, of course. You can study it empirically. So what I was doing here in the parameter scan, when you repeat the simulation many times, what you get is that you can output all of this data, and you can use this to empirically draw the distribution. And I mean, I'm not outputting the values here, but I could have created a report file that puts all these numbers that I'm simulating now. As you can see, it's now the value is such that it's more probable to be on the upper side than on the lower.
Yes.
But you can use this to empirically study the distribution as well. Now, as I said, this model is a very simple one. And it's very simple because I believe people have actually studied this analytically. But in a bigger system, it wouldn't be possible to draw the actual distributions. And therefore, you would do exactly this. You would generate, you repeat. So essentially, we're doing a Monte Carlo simulation where we repeat and we basically find the probability distributions for the various molecules.
Thank you.
So any other question?
Hi, I have a quick question. Does COPASI automatically generate the lower steady state and upper steady state lines that you see on that graph? No.
This was done beforehand, so I can show you how that was done. And that was done studying the deterministic system. So in COPASI is plotting them because it basically is using, I'll show you what it is plotting. We created what we call global quantities. These are parameters, and these parameters are not used for anything. We just determine what is the steady state for the high value and what is the steady state for the low value. Actually, I mentioned them in the opposite case. And therefore, we just added these two lines on the plot. So these lines were numbers we added here in the model. But we could find out these numbers by using the time course. If we go back to the ODE approach, now if we run it, we can study it, the value, right? So the value went up there, and I could also do it by steady state. So this gives me one of them, 247. So I can now add a slider as well. I mean, I could study this with the parameter scan, but a slider is good to show this. I can change the initial concentration of the species, move the slider a little bit further up, and the steady state now shows me 566. So this is how we calculated the numbers we've added there, just so we could plot them on the plot. So this was done by hand. It was not automatically. In fact, there's no way to automatically find all the steady states. You have to do something like this. You have to study different initial conditions to try and see if you have, if you determine those steady states. But then once you've done it, you can run the simulation, and you can also plot the values of the two things.
Thank you.
Okay. Any other questions? So one of the things that you have to have in stochastic simulations, which I will mention now, is that the reactions need to be irreversible. So you'll see in this model, it has the forward reaction and the backward reaction written separately. So that is exactly, this here is exactly the reverse of that. And you could have written the reaction reversibly, so that wouldn't work. COPASI would tell you that it cannot run stochastic simulation with reversible reactions, but it has a way of transforming the reversible reactions into irreversible reactions, and that is done here in the tools. And you find there convert to irreversible. So what COPASI will do is that it will automatically split the reactions, each reaction that is specified as reversible, into two reactions, one for the forward, one for the backward reaction. And that will allow you then to run stochastic simulations. So just before, I mean, I was going to originally planning to show some other examples, but I want to give time for Boris to show you spatial stochastic simulation. So I will just mention a few things. So as I showed, it's very easy to switch from deterministic to stochastic. Now in stochastic, we have a number of algorithms. We have 4 of them here. The first 2 are basically the same. The Gibson and Brook method is exactly the same as the direct method, except that it does loops in a special way that makes it faster when you have a very large number of molecules. But otherwise, those 2 are the same. The tau leap method is an approximate method, so it's no longer using the exact stochastic algorithm of Gillespie, but it is actually calculating, trying to find out for a certain amount of time how many reactions happen there. And therefore, it can jump more than the time that is needed for one reaction. It can jump for every 10 reactions, for example, and that makes things faster. Some algorithms are very slow, and otherwise they become faster with the tau leap. And then there is this stochastic adaptive tau leap, which is basically trying to do a compromise between the tau leap and the direct method. And it's basically just looking for when the tau is large, it uses tau. When it becomes too small, it then switches to doing the tau leap method. So it's an adaptive way, and that might be a good algorithm to try if you have very slow reactions. Then there's a few others down here. I will just point the last one. I don't have time to go into this, but we also have a stochastic differential equation solver. So you can have an ODE system, and you could, in the reaction, add a noise term. And if that noise term is this, which is the square root of the flux of particle flux, then this is what we call a Wiener process. And you have now also a stochastic differential equation that represents the accurate, at least in the Langevin domain, of the reaction. So you could also use that approach. I don't have more time to talk about this. In the question answers times, if there are questions about it, I may be able to answer some of these. But I'll just point it out just so you know that COPASI also allows you to do stochastic differential equations as well. So I will end my presentation here, and I'm now going to stop sharing my screen so that Boris can move on to the spatial stochastic simulations.
All right, thank you, Pedro. Let me share the screen. Okay, so I'm going to tell you about solvers that Virtual Cell provides for solving spatially resolved stochastic models. So let me see if I have. Yeah, so basically what I'm going to do is number one, overview methods. Number two, explain what are the VCell solvers, what the capability of VCell you could use to solve spatially resolved stochastic methods, and a couple of examples. So before I go to actual specific methods for solving stochastic spatially resolved models, I want to just clear up one question. What are we trying to find by solving a stochastic model? The answer to that question is that what we're trying to find is the time-dependent probability distribution of the system. For the well-mixed model, the system is quantified in terms of copy numbers of molecules. And so basically your probability distribution function is defined on the space of all those possible combinations of copy numbers. Now in space, you add spatial dimension, and say even if you discretize space, then the dimensionality of the all possible combinations or all possible outcomes in the stochastic model becomes huge. And although you have just to find one function, which is time-dependent probability distribution, but the domain in which it is defined is so large that solving the differential equation that governs this time-dependent probability distribution is no longer practical for almost any systems. So you have to resort to methods that simulate individual trajectories of the system, individual kind of like experiments. Like you do in real experiments, you have some outcome, then you repeat it, and the outcome may be different due to stochastic noise. And so you do this with the computer. And the tool that allows you to do it is random number generator, which is built now in each computer. And the methods that use that approach is called Monte Carlo simulations. One thing to remember is that when you simulate just one outcome, as Pedro just illustrated, it's not sufficient. You have to do multiple trials. And then you have to statistically process the results and to build some kind of a histograms. And those histograms, not just one histogram, there's a series of histograms as corresponding to different time points. So it becomes a very labor-intensive process. So one comment is that usually stochastic methods, numerical stochastic methods, are conceptually straightforward. But the actual process, first of all, you have to run multiple trials. Second of all, you have to do statistical processing. That whole thing is far from easy. Now, and in particular, when you go to a spatially defined system, that kind of multiplies the logistical problems like infinite number of times. Now, going to methods. There are two basic methods that are used for solving spatially resolved stochastic models. One is called particle-based methods. There are two popular packages. SMOLDIN, that stands for Smoluchowski dynamics, and VCell, that's M stands for Monte Carlo. And the methods based on the so-called reaction diffusion master equation, RDME. And the package is called MesoRD, and it's based on the so-called net subvolume method. So in particle-based methods, the system is defined in terms of spatial locations of each particle, of each molecule, and the state of that molecule. The idea behind the RDME methods is this. First, you split up your space into subvolumes, usually cube subvolumes. And then you treat the subsystem of molecules that happen to be in a particular subvolume like a well-mixed system, and you apply basically the same methods that Pedro just described. Now, so you will have reactions, and diffusion steps are also treated like a reaction step where you destroy molecule in one subvolume and generate and create a molecule in the neighboring subvolume. So your stochastic steps now includes real reactions, and also kind of auxiliary reactions that represent diffusion steps when particle crosses from one subvolume to another subvolume. And actually, the next subvolume method is a general kind of steal the idea of the next reaction method for non-spatial systems. They first throw a die and determine in which volume the reaction will occur. And then they apply the machinery of the next reaction method for that subvolume. I would like to also make this comment that Monte Carlo methods based on random number generation are also used just for purposes of solving purely deterministic problems, such as evaluation of multidimensional integrals, solving actually the problem that can be solved by reaction diffusion equations, simply because it's very conceptually simple. Just throw in particles and follow them. That's what early implementation of VCell were. So they basically solved the problem that could be solved deterministically, but they just prefer to do it using random number generator. Now, a virtual cell offers two different ways of simulating spatial models. In both cases, stochastic spatial models. In both cases, the engine is the Smolden simulator. So it was integrated in the virtual cell for these purposes. So you have to keep in mind that if you want to do it spatial, you have to represent each molecule as an individual particle. And certainly, if the number of particles increases, the simulations become slower. Still, it is practical, I believe, to run these simulations up to, I would say, 500,000 particles. I think it's okay. Still, you can still do it. The first simulator is when you simulate all the molecules, all the species in your system as particles. The second one is a hybrid simulator for cases when some of the species in your system are expressed in very large numbers, and if it was just them, you would be okay to solve it deterministically by solving partial differential equations. But there is a subsystem, say, subsystem of variables that are expressed in low numbers, and the noise due to this small number of molecules actually, due to interactions between the so-called stochastic system and subsystem and deterministic subsystem, actually confers the noise onto the deterministic variables. And you see kind of large fluctuations, stochastic fluctuations in that subsystem too, because the whole system now becomes stochastic. But if you were to use the first method, that would be really inefficient because you will have to, to follow each particle in those, for those variables that are basically expressed in very, very large copy numbers. I would also add that this, as COPASI, VCell also offers a variety of solvers for non-spatial well-mixed stochastic systems. And finally, I will go to a couple of examples. This is the, is in COPASI. So once you set up a model, so if you use the biomodel workspace, and you created model there, you have a choice. I guess, I'm sure Les showed that to you yesterday. You have a choice to treat it either deterministically or stochastically. You can create stochastic applications. This particular example, I used the published Elf-Ehrenberg model. And just as an example, I first created a deterministic application. And then I did stochastic applications for 2 different volumes. Now, when you go to this icon, you will see something like this window. And you will see that the integrator is going to be Smolden, Spatial Stochastic Simulator. All you have to do is specify end time. You also have to specify the timestamp because the Smolden integrator is a fixed time integrator, particle-based fixed time integrator. If you specify the interval that you want to save your results with, and you can either do random seed or you can put whatever seed you want here. Once you fix the seed, it's actually it's a predictable now, the sequence of random numbers, so it's good for testing. Say, if you want to try to say you have a model and then you make changes or something, or you don't remember that you made changes, you can rerun with the same random seed and you will see whether, in that case, if there were no changes, the results should be exactly the same as in the previous, even if it's a stochastic model. And the last example is for this hybrid model. This is a toy, I mean, this part is a toy model of calcium sparks. So you have small number of calcium channels, and they open, close and open stochastically. But with each opening, a huge number of calcium ion will come from, say, sarcoplasmic reticulum into the muscle cell. And although the calcium, free calcium, is expressed in a large number of, I mean, you have very large number of copy numbers, large copy numbers. You can still model the whole system stochastically using this hybrid approach just to show you if it works. This is simulation results with one frequency of opening and closing, and this is with the, you can easily tell that there was much higher frequency. This screenshot is from another model where you have adhesions as represented as a stochastic subsystem of a larger system. And REC and myosin are the molecule, the variables that are treated deterministically. If you have questions, please let me know.
So, excuse me, I have one question.
Okay, who are you? I cannot see.
This is Junhao. I just want to know that, like, how specifically can you enforce this hybrid? Like, do you kind of like just put it as a function and then time it to the rate constant or something?
Okay, so first of all, I should let you know that the description of this method was published in PLOS Computational Biology. I can give you a reference to that. So the idea is this. So when you write the equations for this model, your deterministic variable will be described as a partial differential equation with the source that depends on the stochastic variable. At the same time, when you write the probability distribution for the Poisson processes that describe the stochastic variable, some rates of, you know, transitions from open to closed, say in the simplest possible case, from the open to closed state and vice versa, can be a function of the deterministic variable, the concentration of the deterministic species that describe deterministically at the location of the particle. So now you have this coupled system. The method is, again, so at each time step, it's a fixed time step simulator, and at each time step, you have to update both systems. And for updating the stochastic system, we use Smaldin engine. And for solving the deterministic equation, we use one of the VCell solvers that also fixed time step PDE solver. Because we're going step by step, then basically, even if you have a stochastic component there, at the moment, then when you integrate the deterministic equation, you know that in the particular location you have a source that's working at this moment. So there is nothing unusual for solving then the PDE with this source. At the next time, that source can be closed. wasn't working, but you can still solve that equation at that particular time. And when you update, when you use Smoldean to update your stochastic model, at the same thing. So you solve that, you know, you simulate it because all the rates are now updated and you know what they are. Did they answer your question?
Yes. Maybe another more regarding to the stochastic solver. You said that.
I'm sorry. Let me just add one more thing. As I said,
Yeah.
when you do one sweep, when you do one simulation, it's not sufficient because our purpose is to figure out the probability distribution function. That's the mathematical problem that we are solving using Monte Carlo simulation. For that, you need to run, let's say, at least 1,000 trials and then build histograms over each pixel or over each subvolume in your system for each variable. All right? Okay, go ahead.
Yeah. So then my question is for the stochastic solver. I know that fixed time step maybe not the most efficient one. So does VCell have any kind of like version of like a adaptive time step?
VCell or VCell?
No, VCell.
Well, for spatial simulations, both solvers are fixed time integrators. And you're right, so they may not be entirely efficient when you have like when you have reactions with disparate time scales. However, usually you would think that stochastic subsystem is kind of slow. Because if the transitions between discrete states are very fast, then even if you have only a small number of molecules, they still kind of be approximated not badly deterministically. So let's assume, for the sake of argument, that stochastic system, even there are some discrepancy in frequencies of various stochastic jump processes within the stochastic system, they're still more or less slow. Usually, I mean, at least in those examples that we encounter, the problem is with the deterministic part, where you can have really disparate time scales. In this case, for the deterministic part, our solver is equipped with the option of quasi-steady state approximation when it's appropriate. So you can invoke that, and that would speed up simulations. The problem with the agent-based, I guess, approach, or how you call it, when you also predict when the next reaction will occur, there are actually quite there are problems with synchronization. Some of them logistical, some of them conceptual. So it's you have to weigh the efficiency and the actual problems with the implementation. So at this point, we kind of came to a conclusion that the fixed time integrator is good for the applications that we so far dealt with.
Okay, thank you.
All right. You're welcome. Any other questions? Don't hear anything, I guess. Thank you for listening.
Okay, looks like we have 5 minutes. 5 minutes left.
Use them.
I can use them.
Yeah.
If you stop sharing your screen, I can show something that I had on my slide, so people might.
Yeah, maybe one more question.
Yeah, go ahead.
So in COPASI, you can automatically run a lot of iteration for the stochastic simulation. Is it also possible in VCell?
Well, for the, as I said, virtual cell also offers a variety of methods. Next next reaction method, which is a version by Gibson and Brack, which is pretty efficient, and also hybrid methods. For those methods, yeah, there is an automatic option for running multiple trials, and it will return a histogram to you. As for spatial methods, that's a totally different ballpark game. It's you have to actually talk to us probably, or you have to do it manually or something. But because that generates a lot of data, before using that, yeah, you certainly should run multiple simulations. Don't please lose sight of the fact that one simulation is not enough. And you can use this parameter scan feature in the virtual cell. You can create a parameter like number of trials and say 10 or 100 or something. It will do it for you. But if you want to do it, you know, more or less, to use some automation, you have to contact us and we will work with you.
Okay, thank you.
Mm-hmm.
Okay, so I will just, in the short couple of minutes, if you loaded my slides, you might have seen this very scary last figure. This is to talk about the Lotka-Volterra model. This is a very famous model, comes from the 1910s by Lotka and Volterra, that was on predator-prey interaction. So it could simulate foxes and rabbits. And the foxes, well, the rabbits eat grass and grow and reproduce. That's that reaction here. Two rabbits, this should actually be wrong. It's rabbit plus rabbit. It's actually correct. Rabbits go to two rabbits. This is kind of self-generation, but forget about it. And then the foxes eat the rabbit. So a rabbit plus a fox gives two foxes, and the foxes can die out. So we can look at this model again in the deterministic way. So I'm going to show it here. It's very simple. It only has two species, the rabbits and the foxes. And it only has three reactions, just like I showed there. When we run time course, deterministic time course, we have an oscillation system. And you may have seen this before, the rabbits and the foxes. I mean, as the rabbits increase in number, the foxes start getting more to eat, and they basically all oscillate. And the foxes eat a lot of rabbits, the rabbits go down in numbers, then there's no food for the foxes, they don't reproduce as fast, etc. Then more rabbits, and they enter basically this system. So what happens when we run this stochastically? So let's look at the stochastic version of the rabbits and the foxes and what can happen there. So a few things can happen. As you can see in this one, the peaks are no longer the same when I run the simulation. And here in this case, you have a number of oscillations with different amplitudes, but at some moment, the rabbits die out. The foxes ate too much and there's no more rabbits left. And of course you need rabbits to make more rabbits, but very quickly, so the red ended here, it went to 0. And then very quickly, the foxes also died out because of course there's no more rabbits to eat. So this shows one thing that happens in this case where, again, the stochastic solution is not the same as the deterministic because you have a possibility of extinction. In the ODEs, the numbers never get to 0. They get very, very, very small numbers of rabbits, but that just kills the foxes out, and then the rabbits start growing again, because even though that number is very small, it's not 0. But in the stochastic version, it goes one rabbit, and that last rabbit is eaten by a fox, then there is extinction possible. And if you'll see, this of course depends on the rate constants, but if I run this again, in this case, there was just one extinction, one oscillation in extinction, but there's another possibility, which is that the foxes may also die out. That can happen sometimes. And there you go. When the foxes die out, this model is very bad because it doesn't have the rabbits dying other than being eaten. So now you can see the rabbit population is exploding. It's already down, 8 million, 80 million, 100 million rabbits, because the foxes died out. And now COPASI said there's too many numbers. I cannot run stochastic simulation with more than 120 million. It's too much for me. So this is what can happen as well. So that's another thing, just a very quick example. I will stop here. Yes, the answer to the question about COVID simulation is that yes, we can simulate COVID using actually ODEs or stochastic models. And in fact, I am doing that, but I'm not going to talk about it here because that's too much to put in, and I've already gone now over 2 minutes. So I'm going to stop. But yes, it's possible to do this with COVID, and I hope that there is not going to be any extinction. If there is, I hope the extinction is only of the virus, not of people. And I don't think that would be the case, but certainly we hope that the virus can be extinct. Okay, so I'm done, and I'm going to give it back now to, I believe, Masood.